Hello there, Bio 16. We're going to be covering blood vessels and lymphatic vessels today. This is a figure that we call Blood Vessel Man. He's one of the models that we have in our lab. This is one of the models that we would like for you to study for your lab exam. I will be posting pictures on the Canvas site so you can find those easily. So the circulatory system is a closed system compared to other organisms that have an open circulatory system like insects. Our closed circulatory system allows blood to circulate between both the pulmonary and systemic circuits of the body. Now, what are those? The pulmonary circulation is taking blood to the lungs where oxygen can be added to hemoglobin, or the hemoglobin actually picks up oxygen. So oxygen comes out of the lungs, attaches to hemoglobin, but at the same time, it's possible for lungs to get rid of CO2 here. So the blood circulates around through the lungs and CO2 is eliminated and we breathe it out. So our circulatory system is allowing the regulation of gases in our body. Remember, if you haven't taken physiology yet, you're going to spend a lot of time on this. Our body produces CO2 as a byproduct of our energy systems. And so our metabolism is constantly producing CO2. We use oxygen to make energy, but you'll hear more about that in physiology. So that's a pulmonary circuit, the circuit of blood through the lungs so that oxygen can be absorbed and then taken out to the body through the systemic circuit. Through the systemic circuit, you're going to see the delivery of oxygen to tissues, and CO2 will be entering the systemic circuit here, so it can be circulated out to the pulmonary circuit. We'll see a picture of that in a little bit. Both the systemic and pulmonary circuits are being pumped simultaneously. In other words, blood is flowing through both and the pump is the heart. So both circuits, you're going to have arteries branching from large arteries down to smaller arteries and eventually to arterials and capillaries. Capillaries are where gas exchange is going to occur. We're not going to deal with all those details in this class, that is in physiology, but we are going to talk about how the structure of blood vessels affects gas exchange in tissues. So all gases, and to top it all off, chemical exchange. So like chemicals like sugars and metabolic wastes occur at the level of capillaries, where you have tissues, okay? Ooh, she's going to try to write again. TISS, that's going to be tissues. Sending waste to the blood. Oh boy. And the blood at the same time is going to be sending nutrients to the tissues, all right? So you have this exchange of waste products into blood and blood supplying nutrients. So let's start by looking at what the blood vessels look like. We're going to start with the largest, the arteries. The largest arteries, or the largest artery in the body, is called the aorta. And so we have the pulmonary arteries and the aorta themselves that leave the heart. The arteries branch many, many times into smaller arteries called arterials. So you have like a giant, almost fire hose. It's not literally a fire hose size in the human body, but that's the largest blood vessel. It would be like the big fire hose would be the aorta. The aorta would then branch eventually down to arterials, smaller hoses. The arterials are where blood pressure is regulated. Arterials then branch again into smaller arterials where blood pressure can be regulated there as well. And eventually the smallest arterials branch into capillaries. Capillaries is where you're going to see chemical, nutrient, and waste exchange and gas exchange happening at the level of tissues and circulation. The capillaries then themselves merge into really small veins called venules. The venules that then merge into larger veins, and then eventually the veins circulate back around to the heart. And we'll show you a few pictures of that in a few minutes. So let's start with blood vessel histology. So remember, histology is the field that studies the internal structure and the tissues of a given organ. So the walls of the blood vessels have 3 layers. Starting at the beginning is the tunica intima. That's supposed to be the number 1. The tunica intima here is the internal region. This is the internal region of a blood vessel. So inside of this blood vessel, you would find, for example, this is my example of red blood cells circulating around. Red blood cells would actually be bigger than this. The tunica intima is basically an endothelial lining. So right here, you'll see 1, 2, 3 endothelial cells, which are literally squamous epithelial cells. So each one of these little regions here is a single squamous epithelial cell with some basement membrane holding it onto these elastic connective tissue fibers right here. So the yellow or kind of, I don't know, flesh colored here is connective tissue, and these endothelial cells are attached to that. Just so you know, these endothelial cells you'll find all throughout the circulatory system. Endothelial cells line the inside of the heart. They continue out through the aorta, out through the arterials, all the way to capillary beds, venules, and veins. So you basically have this internal tunica intima that lines the entire circulatory system. The tunica media here is going to be our smooth muscle. Okay, so this is layer number 2. Isn't it funny that I can make a 2 more easily than a 1? Haha. So the tunica media is the thickest region of any artery, and the tunica media consists of lots and lots of smooth muscle and some connective tissue. That's what these little blue lines are here, right here. Okay, these little blue lines are bands of elastin. And basically, the tunica media is going to be the smooth muscle that contracts and regulates the diameter of an arterial or of an artery, basically. And so what happens is when the artery constricts, then the blood pressure goes up. Basically, you've constricted down the diameter of the blood vessel, or the size of the lumen here, okay? So as you make the hose smaller on the inside, blood pressure goes up. Dilation allows blood pressure to go down. So basically, the tunica media is going to be controlling vasoconstriction and vasodilation. And then the tunica externa, or it's also called the tunica adventiva, is made up of lots and lots of connective tissue. So here's layer number 3, tunica externa. Collagen, lots of collagen. This is a protective layer, some elastin to help maintain stretchiness in the blood vessels here. So one of the things to keep in mind is that the largest arteries have to be very, very flexible, very stretchy, because every time the heart ejects blood out of the left ventricle, basically a giant lump of blood is traveling through the arteries. That lump of blood travels as kind of a big lump, and that causes stretching of the arteries. And so a lot of elastin is necessary for pliability and compliance, but also lots of collagen to keep the strength of these really stretchy, very resilient blood vessels. Over here was our artery. I'm trying to find my pointer. Here's our artery, number 1. The veins on the bottom, this is a vein here. When you compare the structure of a vein to an artery, you still have a tunica intima made of the squamous epithelial cells, a tunica media, but you're going to see that the smooth muscle, so here's layer 1, layer 2, the smooth muscle layer is much thinner, so there's a lot less control of blood pressure at the level of the veins and the venules. The tunica externa, over here, has a lot of collagen, but not as much elastin. The reason being is that these veins aren't as stretchy. They don't need to be as compliant, because by the time blood gets to the veins, blood pressure is actually very low. That's something that you're going to be learning about in physiology, and I'm sorry here. Tunica externa is layer number 3. So compare the differences and similarities between arteries and veins for your lecture and lab exam. So how can you tell the difference when you look at a slide? It turns out that veins look very, very kind of open and wide. You can see that the size of the blood vessel, the tunics here, are quite a bit different in arteries versus veins. So here's the depth of the blood vessel wall in a vein. Here's the depth of a blood vessel wall in an artery. Much, much more tunica media. So here's the tunica media in an artery. Here's the tunica media in a vein. Way bigger in an artery. So artery walls are a heck of a lot thicker. When you cross-section them, you can almost always find an artery next to a vein. It's not a rule, but you oftentimes find them traveling together. And arteries usually look very circular in cross-section, whereas the veins look kind of floppy or kind of oval shaped. This is because veins are much wider. You'll hear much more about that in physiology, but basically what happens in veins is that we have a bunch of blood that ends up getting just kind of stored in veins while it's being transported back to the heart. So here are a description of arteries, 3 tunic layers described: the internal elastic membrane, a really thick layer in the tunica intima that the endothelial cells are attached to. Arteries expand because a big lump of incoming blood comes from the left ventricle. There are 2 major types of arteries that you find in the body. Number 1 are the elastic arteries. These arteries are closest to the heart. They're the most elastic. They have a little less smooth muscle, and the reason why they're more elastic and they have a little less smooth muscle, it's believed, is that they have to basically handle lots of high pressure blood coming from the left ventricle. So this would be basically like the aorta. Muscular arteries, number 2 here. The muscular arteries are quite a bit smaller. So elastic arteries are about 1 inch in diameter, pretty big. The muscular arteries are about, 0.15 of an inch to maybe, 1/5 of an inch in diameter. These have a lot more smooth muscle. The tunica media is much thicker here. A little bit less elastic tissue, and that's because these are the blood vessels or the arteries that are controlling your blood pressure. And so it ensures that your body has enough blood flowing out to tissues at a fast enough rate, so this diameter is fine, takes enough blood out there, but this is the hose that's adjusting to ensure that blood pressure is consistent while you're doing what you need to do throughout the day. The arterials are much smaller, thinner walls. There's a much more poorly defined tunica externa. The outside, there's still connective tissue there, but the blood pressure is lower here. And so what you have is a situation where you don't have to worry about as much blood going through these arteries, so they don't have as much tunica externa there. They do have tunica media, and you're typically going to find these arterials inside of organs. So the organ tissue itself helps protect the outside of the blood vessel. You have lots of vasodilation and vasoconstriction regulating how much blood is going to the organ. This is autonomically controlled. So if you go back and think about the autonomic nervous system, fight or flight, rest or digest, you're going to have a lot of vasodilation under rest and digest. Parasympathetic. Lots of vasoconstriction under sympathetic control, depending on the organ. Capillaries themselves, like I said, are mostly that endothelial, squamous epithelial cell layer. We call it endothelium, plus the basement membrane holding all those cells in place. They are usually found near almost every living cell, depending on the thickness of the tissue. And the vessels themselves, the capillary vessels, have gaps between the individual squamous epithelial cells, allowing chemical diffusion and gas exchange to occur between the bloodstream and the tissues. Some capillaries are called fenestrated. You're going to find fenestrated capillaries in the kidneys. This allows much larger molecules to sometimes escape out into other circulation, but we'll talk about that later.